Neurons use many different second messengers as intracellular signals. Here we will discuss the calcium ion, which is perhaps the most common intracellular messenger in neurons. In this example, a ligand binds to a calcium channel, causing it to open and let calcium ions into the cell. In another example, a voltage change across the plasma membrane causes a voltage-sensitive calcium channel to open. This transient rise in cytoplasmic calcium concentration transmits information within the cell. The rise in calcium allows calcium ions to bind to a large number of calcium-binding proteins that serve as molecular targets, such as calmodulin, which is abundant in the cytosol of all cells. Binding of calcium to calmodulin activates this protein. Calmodulin then activates further signaling pathways by binding to downstream targets such as protein kinases. In order for a rise in intracellular calcium to be sensed by the cell must carefully maintain resting levels of calcium. Ordinarily, the calcium concentration in the cytosol is low, about 100 nanomolar, which is the same as 1 × 10 to the -7 molar. The concentration of calcium ions outside neurons is about a millimolar, or 1 × 10 to the -3 molar. This steep gradient is maintained by a number of mechanisms. Proteins in the plasma membrane transport calcium out of the cell. One type is an ATPase called a calcium pump. The energy from ATP hydrolysis is used to pump calcium up its concentration gradient, while hydrogen ions are counter-transported. Another means of maintaining the gradient is by the action of a sodium-calcium exchanger. This exchanger uses the energy from a concentration gradient of sodium ions across the membrane, exchanging sodium ions for calcium. When calcium levels rise within the cell, a variety of calcium-binding proteins, in addition to calmodulin, bind to the calcium. One other example is calbindin, which serves as a calcium ion buffer. It reversibly binds calcium and thus blunts the magnitude and kinetics of calcium signals within neurons. Calcium ions are also pumped into the cell's mitochondria and endoplasmic reticulum. The endoplasmic reticulum uses a calcium pump similar to that in the plasma membrane. As calcium levels fall, the signaling systems return to their resting state. Through the action of the pumps, the ER and mitochondria can serve as storage depots of calcium ions that are later released to participate in signaling events. For example, channels in the ER allow calcium to be released from the interior of the endoplasmic reticulum into the cytosol. One such channel is the inositol triphosphate, or IP3, receptor, which opens when another second messenger, IP3, binds to it. Another calcium-releasing channel is the ryanodine receptor, named after a drug that binds to and partially opens these receptors. Among the biological signals that activate ryanodine receptors are cytoplasmic calcium and, at least in muscle cells, depolarization of the plasma membrane. In these ways, calcium ion concentrations are modulated and used to convey information in the cell.